With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and microcap data place. Welcome to the virtual Roadshow series presented by Channel Check. This Roadshow features Bedu Addo, CEO, Lauren Wood, CMO, and Seth Van Voorhees, CFO of PDS Biotechnology, NASDAQ ticker symbol PDSB. Following a formal presentation, they are joined by LeBoyer, Senior Research Analyst for Noble Capital Markets, a FINRA-licensed, SEC-registered broker-dealer, for a Q&A session featuring questions that were submitted by the live audience. You can find Noble's research on PDS Biotechnology on channelcheck.com or by clicking the link in the description. With that, I am pleased to present Frank, Lauren, and Seth.
Chris, thank you very much for the introduction, and thanks to Noble Capital Markets for having us participate in the virtual Roadshow series. To the viewers, please note that this presentation does contain forward-looking statements. Please ensure that you are familiar with all the risk factors associated with the company as filed with the SEC. The PDS Biotechnology is a mid-clinical stage biopharmaceutical company. We currently have 3 ongoing phase 2 human clinical trials to study the company's lead immunotherapy called PDS0101. PDS Biotech has established important validating clinical partnerships with some of the most highly regarded oncology institutions, including Merck, the National Cancer Institute, and MD Anderson Cancer Center. PDS Biotech also has no debt and reported approximately $29 million in cash as of our last filing on December the 31st, 2020. Now, we announced in February that our phase 2 human clinical trial in advanced HPV-associated cancers in patients whose cancer had returned or continued to spread after treatment had achieved its initial threshold of tumor shrinkage in 3 or more of the first 8 patients, therefore allowing for recruitment of the full cohort of 45 patients. Now, as Dr. Wood will explain later, this threshold represents about a 30% improvement over what has been reported to be the most effective therapy in this patient population to date. We are also working with our 2 Brazilian partners as a consortium to develop and commercialize a novel Versamune-based T-cell activating COVID-19 vaccine in Latin America. We announced in March that the consortium has been granted up to approximately $60 million by the Brazilian Ministry of Science, Technology and Innovation, the MCTI, to develop the commercial vaccine. The versatile Versamune technology platform and products are protected by several composition and application patents issued in several countries and valid through the mid-2030s. To date, the most significant limitation to effective cancer immunotherapy has been ineffective induction of tumor-attacking killer T cells, also known as CD8 T cells, that can be produced within the body with both adequate potency and quantity. At PDS Biotech, we are developing a pipeline of cancer immunotherapies and novel vaccines based on our proprietary Versamune technology platform that has been engineered and demonstrated to generate powerful killer T cell responses. I'll discuss the impact of this characteristic of Versamune on anti-tumor efficacy as we review our data. Now, in addition to powerful in vivo CD8 T cell responses, the Versamune technology, or the Versamune cancer immunotherapies, also possess the following critical characteristics. First, the ability to induce long-term memory T cell responses, which results in long-lasting efficacy and robustness of the anti-tumor effect. Secondly, a unique combination of potency and safety without the systemic toxicities typical of immunotherapy and cancer treatment. And third, the versatility based on the safety of the technology or the products to allow for combinations with multiple or various cancer therapies. So here on the slide, I show the company's robust pipeline. In each product, Versamune is combined with a different tumor or infectious disease-related protein, which is specific to the particular cancer or infectious disease we're seeking to treat or prevent. Versamune trains the immune system to recognize that disease-specific protein as a foreign agent and also activates our T cells to attack the diseased cells. Our lead program, PDS-0101, addresses various HPV-associated tumors, including anal cancer, cervical cancer, head and neck cancer, vaginal and vulvar cancers, and is being studied in 3 phase 2 human clinical trials. With our infectious disease programs, our universal flu vaccine studies are funded by a grant from the NIAID, and our COVID-19 vaccine program, as I mentioned, is fully funded through commercial development by the MCTI Brazil. So now before we move on to the technology, the clinical trials and upcoming milestones, I would like to introduce you to the company's senior executive management team and to provide short backgrounds on our relevant experience. I am a chemical engineer by training with my PhD in pharmaceutical drug development. My career my early career actually was as a drug development scientist at companies such as the Liposome Company and Schering-Plough, which is now part of Merck. As a drug development scientist, I worked on the development of several currently marketed drug products. At Cardinal Health, I was hired to start up and head up the company's East Coast biotech drug development operations and saw all business, profit and loss, and drug development operations. Now, based on the success at Cardinal Health, I was hired to the senior executive management team of KBI Biopharma as one of the founding executives to develop, implement, and oversee the company's operations. KBI Biopharma today is a very successful protein drug development company. I was at KBI Biopharma until the scientific founder of PDS Biotech recruited me to take the very early stage technology concept and build a successful cancer immunotherapy company. We started early stage drug development operations 12 to 13 years ago, and I have been responsible for overseeing the company's operations since inception of the company. I'll hand over to Seth to provide you with a short bio.
Good afternoon, everyone. I have a PhD in chemistry, and earlier in my career, I worked as an investment banker, helping companies like PDS raise capital to fund their operations. Over the last 15 years, I've worked as a CFO of American Pacific, a manufacturer of APIs for cancer treatment, as well as for Research Frontiers with a focus on nanoparticle technology. And I joined PDS in January 1st of this year. Lauren?
Okay. Lauren.
Thanks, Seth, and good afternoon, everyone. I'm Lauren Wood. I'm a physician scientist and actually a trained allergist immunologist with training in both internal medicine and pediatrics. I have a 30-year extensive career at the NIH, specifically at the National Cancer Institute, involved in investigating across all phases of development, both antiretroviral agents in the infectious disease space early in my career, as well as immunomodulatory agents for both cancer and HIV. Frank managed to talk me out of retirement after 29 years at the NIH, and I've been the Chief Medical Officer for PDS Bio since February of 2019.
Okay, thanks, Seth and Lauren. Well, our Chief Scientific Officer, Dr. Greg Kahn, is not here with us today, but Dr. Greg Kahn also brings a wealth of experience gained at successful companies such as Merck, Regeneron, and Diosynth. His early work led to the successful anti-cancer drug Avastin, and Greg has taken drugs through the entire drug development cycle. Okay, so moving on to the technology. our Versamune technology platform is designed to promote activation of T cells within our body. Now, T cells differ significantly in both type and potency. Therefore, it's important to generate an effective anti-tumor immune response. The immunotherapy must induce the right type of killer T cell in the right quantity and also with the necessary killing potency. So right type, right quantity, and right potency. All three are critical to induce an effective anti-tumor immune response. So on the left-hand side of this slide, I have a bar chart showing the number of killer T cells induced by various clinical stage technologies. The dark green bars at the bottom represent a type of T cell established to be the most potent type of killer T cell. Now, this study shows that the Versamune-based product induces a significantly higher quantity of T cells, but also very importantly, a much higher quality of killer T cell. Now, the effect of this immunological characteristic of Versamune on anti-tumor efficacy is shown on the preclinical tumor regression plot on the right. So here, the tumor size is on the y-axis. In the untreated animals, the gray line, we see rapid and aggressive tumor growth. Now, the black line shows the results when the protein that is specific to these HPV-associated cancers, HPV-16, is administered. And as you can see, upon administration of this tumor-specific protein, there is a complete lack of efficacy. Now, the blue line that I show over here is representative, and this is what we obtained using the clinical adjuvant GM-CSF. This data here, this line is representative of the data reported by the leaders in the field. So what we see here is that with multiple doses, an immune response is generated, or an immune response results, which leads to a slowing down of tumor growth. But the cancer continues to progress and the animals or the patient live a little bit longer. So that is the state of immunotherapy today. Now let's take a look at PDS-01. Represent now formulated with Versamune and administered in the exact same dose as before. Now, what we see here is that after a single dose, Versamune's unique ability to present the protein into the correct immunological processing pathway and T cell presentation pathways, as well as the ability to activate the right immunological signaling mechanisms, results in the induction within the body of large numbers of high-quality tumor-attacking killer T cells. And therefore, what we see here is that as a result, after a single dose, by day 35, we see complete elimination of the tumor. Now, this study has been replicated in multiple independent expert labs several times. A very, very consistent result here. Now, what's also very interesting here, also very important to note, is that after leaving these tumor-free animals for 60 days, coming back and re-challenging them with the tumor cells, 100% of these animals were fully protected against tumor reestablishment. This means that in addition to the strong killer T cell response, we've also induced a memory killer T cell response that continues to protect against future disease. Now, this characteristic of Versamune and its ability to generate powerful antiviral killer T cell responses and memory T cell responses also becomes very important when we talk about our novel class of infectious disease vaccines. This concept is being applied to our novel COVID-19 and universal flu vaccines. Now, this slide I show here summarizes the key characteristics of a safe and effective immunotherapy and clearly shows how differentiated the Versamune-based products are from other approaches in possessing each of the key critical characteristics. Okay, so what I'm going to do now is I'm going to hand over to Dr. Wood to provide an overview of our ongoing clinical programs.
Thank you, Frank. Our strategy in developing PDS01 is to deliver it in combination with established therapies for rapid proof of concept, demonstration of activity, and risk mitigation. As Frank highlighted, we believe that one of the inherent values in the Versamune platform is its unique safety profile. We are delivering it in combination with FDA-approved standard of care, with pembrolizumab, also known as Keytruda, in the first-line treatment of recurrent metastatic head and neck cancer, for which pembrolizumab is an approved indication. We're also delivering it with standard of care chemoradiation therapy in women with locally advanced cervical cancer. Again, the standard of care chemoradiation treatment representing the treatment for this population. We're also delivering PDS0101 in combination with two other investigational immuno-oncology agents owned by EMD Serono in HPV-associated advanced recurrent metastatic cancers. This includes a bifunctional second-generation checkpoint inhibitor, M7824, which targets both TGF-beta as well as PD-L1, and a tumor-targeting cytokine called NHS-IL12. This phase 2 investigator-led study by Dr. Julian Strauss, Jeff Schlom, and James Gulley of the National Cancer Institute is evaluating the triple combination of these three agents, PDS0101, M7824, and NHS-IL12. We know that immunotherapy has become the fourth cornerstone treatment for cancer therapy. The problem and the limitations of immunotherapy, specifically checkpoint inhibitors, is that we know that only a minority of patients respond to them, and that response is limited in patients who really lack a tumor-targeting immune response. And that is the rationale driving this triple combination. Again, it's three agents, M7824, NHS-IL12, and PDS0101. The primary study objectives are to examine the objective response rate in checkpoint inhibitor naive patients. We also have an exploratory objective of examining this triple combination in patients who have failed prior checkpoint inhibitor therapy and are checkpoint refractory. And this is a population of huge unmet medical need because, again, while the responses to checkpoint inhibitor therapy can be very impressive and very durable, they still only work in a minority of patients. As Frank noted earlier, we announced earlier this year the fact that we had received notification that at least 3 or more objective responses were observed in the first 8 patients enrolled in the study, triggering expansion to the full enrollment of 45 patients. We anticipate that trial will be completed in the first quarter of 2022 and expect to be able to announce more advanced data in this population by the end of this quarter. Next slide. This slide is just to highlight for you the importance of translational science, which undergirds everything that we do at PDS Bio. This triple combination was actually investigated in the same preclinical tumor model that Frank had shown you resulted in dramatic regression with PDS0101 alone. This tumor model is one in which PDS0101 was delivered with either M7824 or NHS-IL12 as a dual combination, and then also explored as a triple combination. The thing that is the takeaway message that everyone can see is in the top panel, you see some evidence of tumor infiltration. However, when you deliver PDS0101 plus M7824 and NHS-IL12, the triple cytokine combination, agent combination that we are studying in the phase 2 trial, you can see lots of pink and green dots, red and green dots that represent tumor-specific CD8 killer T cells, as well as CD4 helper T cells. The takeaway message is that you see a dramatic induction of these tumor-infiltrating lymphocytes, which we know are necessary to optimize the function and the outcome of checkpoint inhibitors. Next slide. PDS also has our own sponsored phase 2 trial evaluating the combination of PDS-0101 and Keytruda in the first-line treatment of HPV-associated recurrent metastatic head and neck cancer. This trial is known as Versatile-002. We deliver Keytruda, standard of care, in combination with PDS-0101. And again, the scientific rationale is to deliver these two agents together to augment the ability of PDS-0101 to induce tumor-specific CD8 infiltrating tumor cells in patients, as well as deliver a checkpoint inhibitor that takes the brakes off the immune system and allows optimal anti-tumor responses. Our study goals are examining objective response rates and overall survival, as well as duration of response and progression-free survival. Specifically, what we're looking to do is to move the needle. We know that on average, approximately 20% or 1 in 5 patients responds to Keytruda monotherapy as an agent in this disease indication. Our goal is to move the needle from 1 in 5 patients responding to 1 in 3 patients responding, or approximately 33% of patients with the combination. We anticipate having preliminary data the latter half of this year at the end of Q4 2021, early Q1 2022, looking at objective response rates. We also have a formal interim efficacy analysis planned in the first 38 patients. Next slide. Finally, our third trial is a phase 2 study done in collaboration with investigators at the MD Cancer Institution in Houston, Texas. This is headed by Dr. Ann Klopp and Drs. Olsi Giishi, examining PDS-0101 with standard of care chemoradiation in women with locally advanced bulky tumors. We know that the standard of care delivered within 58 days involves the delivery of both chemo and radiation therapy. Importantly, again, hearkening back to translational science that drives what we do in our human clinical trials, it was noted by Dr. Klopp and her colleagues that individuals receiving standard of care treatment had an induction of HPV-specific responses during the course of their standard chemoradiation therapy. Importantly, those who had significant increases in these HPV-specific responses actually had better clinical outcomes. Hence, the rationale for delivering the combination is to deliver at least 1 dose of PDS-0101 prior to the initiation of chemoradiation therapy and deliver an additional 4 doses during the subsequent course of treatment. Again, we're looking at safety of the combination as well as the rate of tumor regression in patients receiving these combination therapies. We anticipate that we'll have preliminary data from this trial at the end of Q4 2021 and early Q 2022. Important accepted radiographic responses that are early predictors of longer-term disease-free survival and progression-free survival in this disease are the PET-CT at 6 months, as well as the rate of tumor volume reduction by MRI at 30 to 40 days following the start of treatment. Next slide. So again, These three studies that I've highlighted for you are really designed to demonstrate the efficacy and, importantly, the broad applicability of PDS0101 in the Versamune T-cell activating platform. There is the potential to treat all types of HPV-related cancer. Our focus with Versatile-002 is head and neck. With our MD Anderson trial, it is cervical cancer. With the NCI trial, we know that there are individuals with multiple HPV-related cancer, not only cervical and head and neck, but also including anal, vulval, vaginal, rectal cancers. There is the potential with these combinations to enhance the anti-tumor and anti-cancer efficacy of the various treatments. Again, we believe one of the unique values of Versamune is its safety profile and exceptional tolerability that really allows it to be combined with either standard of care or investigational therapies that leads to enhanced and potentially synergistic anti-tumor toxicity and efficacy, but without synergistic toxicities that are often seen when you throw multiple chemotherapy or even multiple immunotherapy agents together. As Frank highlighted, there's also potential applications beyond oncology because the specificity of Versamune is that you have induction of an antigen-specific tumor targeting T-cell response. And importantly, as an immunologist, I love to say that an appropriate T-cell response, a high quality, high quantity potent response, is an immune response that keeps on giving. Why? Because it establishes immune memory. It's easy for us to shrink tumors and even make them go away. The problem is that we know in patients who are resisting cancer, it is recurrent metastatic disease that ends up killing the patients. And you need long-term immune memory to be able to eliminate micrometastatic disease that's not visible on imaging, as well as prevent the recurrence of tumor. We know that PDS0203, our SARS-CoV-2 platform that couples SARS-CoV-2 antigens with Versamune, is capable not only of inducing a high quality and quantity of neutralizing antibody responses, we also have documented that the same polyfunctional potent T cells can be induced with the Versamune platform to SARS-CoV-2 antigens. Importantly, we are designing this platform so that it will recognize conserved structural antigens and hopefully provide long-term immune T-cell memory as well as antibody responses to SARS-CoV-2. There's broad potential for additional pipeline and additional partnerships with our successful phase 2 studies of PDS0101 and PDS0203 because we are targeting other important tumor antigens. Next slide. I'll go on to PDS0203 and our preclinical studies that are leading towards its human development and clinical translation. Again, I highlighted for you that our vaccine candidate is able to induce a really maximal breadth of response, not only a humoral antibody response, but also an antigen-specific CD8 T-cell response. On the left-hand side is data that was recently published late in 2020 by a collaborating group at Mount Sinai Icahn School of Medicine that looked at their SARS-CoV-2 Newcastle disease virus antigen delivered alone and in combination with either Versamune or another adjuvant known to induce high levels of antibodies, Adavox. The key thing that you can take away is that even with very low doses of antigen, Versamune was associated with potent antibody responses as well as dose sparing of antigen, a very important quality in the context of pandemic vaccine preparation. On the right-hand side of the slide, what you'll see is Versamune co-delivered in combination with either the full spike protein in dark navy on the far right, or a component of the spike protein S1, which also contains the receptor binding domain that is necessary to generate the SARS-CoV-2 specific antibody responses that are highlighted on the left-hand side of the slide. What you see is that when you deliver either of these proteins, either the full length or only a portion of it, when you deliver it alone, you see absolutely no induction of CD8 T cell specific responses. However, with both spike S1 as well as the full length protein, you see very significant induction of T cells when these protein antigens are delivered with Versamune. Next slide. So we have never been in the COVID vaccine race to be the first ones out of the gate. Our goal has been always to be able to harness the power and the potency of the Versamune technology platform to induce antigen-specific CD8 T cell responses, as well as long-term memory as our primary goals for generating our platform. As a consequence, we believe that the demonstrated induction of CD8 killer T cells, as well as CD4 T cells, I did not present that data in this deck, but we do have it. And we also have similar data showing that both these CD8 and CD4 T cells are polyfunctional, similar to what Frank showed you earlier in the presentation, are T cells that can target less variable components of the SARS-CoV-19 virus and may be effective potentially against the continuous emerging circulating variants of concern. We also importantly feel that in addition to induction of neutralizing antibodies, we also see this induction of T cell immunity, which again has the potential to provide long-lasting virus-specific immunity and memory to SARS-CoV-2. It is emerging continually the importance of the role of T cells in controlling not only SARS-CoV-2 infection, but also in protection against infection. We also believe, again, with the safety profile that we've demonstrated, that there's a high potential for safety. It's a subunit vaccine. It does not require the use of attenuated, replication-incompetent viruses or traditional adjuvants. Again, we've seen no serious adverse dose-limiting reactions with the Versamune platform studied to date. Next slide. Looking forward, I'll hand this now over to Seth.
Thanks, Lauren. So when thinking about the market opportunity that we're looking at, the products that Lauren was just describing uses a single antigen based on HPV-16, and that market alone is quite significant. There are 43,000 patients in the US alone that have HP-associated cancers. And when you look at the average cost for immunotherapy treatment of those cancers, it's approximately $120,000 annually. The three clinical trials that we discussed today deal with the vast majority of the indications related to HPV-16. So as you can see, this is a large market that is growing. Frank, if you could turn to the next slide. Moving from oncology indications to our COVID market opportunity, what has been described today is that our development effort as part of consortium has received funding of $60 million from the Brazilian government to support all three phases of the clinical trials for commercialization of a COVID vaccine. In addition to the clinical trials, it also will support the clinical manufacturing scale-up of that product. So our phase 1/2 study relating to this product is expected to begin in the second or third quarter of this year, pending approval by Anvisa, which is the Brazilian regulatory agency. Pending the results of this study, PDS-0203 could be commercially available as early as 2023. The initial market for a COVID vaccine is in Latin America is quite large. Latin America contains 650 million people, and Brazil alone represents about a third of that, being about 213 million people. Based on public information of commercial COVID vaccine pricing, the average dose for a 2-dose regimen is between $20 and $74. So you can see this market opportunity could be quite significant if we are successful. Frank, if you could turn to the next slide. When thinking about our technology platform, it is quite flexible, and to the extent that we are successful, could open up a variety of opportunities for us. We are currently working with 4 antigens with our Versamune technology platform, but we have identified 75 antigens that are potentially suitable and could be explored in the future. In addition to our oncology efforts, when looking at our infectious disease efforts, as mentioned, we have an active COVID-19 program, and we also have one focused on universal influenza. And when looking at the future, we could take various antigens and look at other indications besides the ones that we're currently working on, which should open up licensing and other business development opportunities for our company. Frank, next slide. When looking at the milestones, the 3 clinical trials that we discussed today, we expect interim results to be released for the NCI trial in the 2nd to 3rd quarter of this year. Furthermore, we expect the 2 other trials to release interim data between the 4th quarter of this year and sometime in the 1st half of next year. And with regard to the COVID-19 commercialization program, we expect a rapid phase 1/2 program to get underway sometime in the 2nd or 3rd quarter of this year that could ultimately result in a commercialization by 2023 if successful. Frank, if you could move to the next slide. And why don't I hand this back to you, Frank, to take on this slide.
Okay, thanks a lot, Seth. So to summarize, our early clinical data as well as preclinical data suggest potentially superior efficacy of PDS0101 and the Versamune-based products. There is strong potential for near-term value appreciation based on the expectation of more mature data from the National Cancer Institute-led trial in advanced HPV-associated cancers later this quarter. We also see our partnerships with top immuno-oncology institutions for each of our 3 ongoing phase 2 clinical trials as strong validation of our science, technology, and target indications. Also, our ability and decision to combine PDS0101 with standard of care in 2 of the ongoing clinical studies, we believe presents a potentially rapid path to commercialization of PDS0101 if the combination is demonstrated to be superior to the current standard of care. We also believe that this combination strategy with standard of care, as well as our strategy of targeting multiple HPV-associated cancers after commercialization, will facilitate rapid adoption of PDS0101 by oncologists. With our COVID-19 program, barring any unforeseen events, we are hopeful that an effective and broadly protecting Versamune-based vaccine could be commercially available in Brazil by 2023. Thank you very much again for the opportunity to present PDS Biotech today, and we will be happy to answer any questions that the audience may have.
Thank you, Dr. Bedu Addo, and thank all of you for those presentations. I'm Robert LeBoyer, the Senior Biotechnology Analyst at Noble Capital and the analyst who covers PDS Biotechnology. Next, we'd like to take some questions from the audience, but before we do, one question from me is that just to put immuno-oncology in very plain language terms, the current drugs like Keytruda are aimed at stopping the tumor from being recognized by the current drugs like Keytruda, and the PD-1 interaction is the basis for many of them. Based on that, would it be correct to say that the immuno-oncology drugs are eliminating the defense and Versamune is increasing the offense after the castle walls have been breached? You're sending in the cavalry and the artillery and then the infantry to kill the tumor?
Right. So Lauren, I can start and then you can jump in.
Okay.
But I think that's a really good description. So the way we very often describe the checkpoint inhibitors is the fact that they more or less expose the tumor or decamouflage the tumor. So that one of the mechanisms by which tumors continue to evade and grow in the body and to survive is to actually evade the immune system surveillance. And so what checkpoint inhibitors do is more or less expose them or decamouflage them, make them much more visible to the immune system as one of the key mechanisms. What the Versamune technology does is it actually it recruits the army, trains the army to recognize that tumor as a foreign agent, and then provides the trained army with the ammunition to then go out, target, and kill the exposed tumors. Right? So we have the enemy that's hiding. The checkpoint inhibitors can expose that hiding army. But then if the army is exposed, or if the enemy is exposed, but there's no one to really recognize that that is actually an enemy, the tumors continue to grow. And that's what happens in the majority of patients who are treated with checkpoint inhibitors. And so what our technology then does is says, okay, here T cells, this thing over here is a foreign agent. Let's recruit a large number of T cells and train them to recognize that particular entity as a foreign agent. And then let's provide them with the powerful ammunition, now that they know who the enemy is, to go out and effectively kill that enemy. And Versamune also has a mechanism by which it also exposes the tumor. So we believe that with these combinations, exposing the tumor by the mechanism of checkpoint inhibitors, exposing them further by Versamune's technology or mechanism, and then using Versamune to attack them, provides the best of both worlds and could provide the most significant clinical benefit to the patients. And Lauren, would you like to add anything to that?
Well, I love the military analogy because I think everybody can understand it, as you started, Robert, and as Frank expounded upon. And so the only thing I'd like to highlight is that we all know in a war, the soldiers have to actually make inroads into enemy territory. Ultimately, you got to get at the heart of where the enemy strongholds are. So as checkpoint inhibitors decamouflage the enemy and make it visible, as PDS-0101 arms literally a massive army of soldiers, but armed importantly, not only with the ability to recognize that tumor and attack it and kill it, importantly, they're potently armed. So they're not, their weapons aren't shooting blanks. They're really able to do the job and then actually infiltrate into that enemy territory, which we know further enhances the activity of the tumor being decamouflaged.
Right, and this is described on slide 8 in the presentation.
That's correct.
Okay.
Oh, and as the immunologist, one more thing. The soldiers actually also have immune memory, so they don't forget what the enemy looks like. So even if you wipe out that major stronghold, that's the beauty of an immune response that keeps on giving. Because if there are little satellite enemy camps, or if the enemy comes back over the hill, the trained soldiers are armed, ready to fire and recognize that enemy.
All right, now let's take a question from the audience.
Okay.
And there seem to be some questions about the COVID program. The first one is, does the company plan to submit an emergency use application with the FDA if the clinical trials yield positive results?
I can take that one. So currently, the focus is on Brazil. So our partner Farmacore is responsible for managing and performing the clinical trials in Brazil. Specifically, they are working with Anvisa and also working directly with the Brazilian government or the MCTI with the goal of getting this commercialized. So our Brazilian partners are really responsible for driving that program forward in Brazil. Now, in terms of the potential use and authorization of those vaccines, currently, the focus has been on Brazil. But definitely, if this vaccine is successful in the clinical trials, as we hope and as we anticipate, we will definitely have those discussions as to where else we could definitely apply those vaccines. But currently, the focus is on Brazil and Latin America.
And do you have a time frame or a target date for emergency use or full approval?
No, we do not. We do not. We know that there have been some Farmacore has mentioned the potential that they could be seeking emergency use approval authorization in Brazil sometime in 2022. However, we have not had any such discussions yet. Those are discussions that Farmacore has mentioned that they have or would be potentially be initiating with the Brazilian agencies in the very near future.
Sure, that's a difficult one to predict.
Correct.
And could you discuss the royalty structure between Farmacore and PDS?
We license the technology to Farmacore, and that's based on a 50-50% royalty split. Since that license agreement was formed, we also expanded that venture to include other consortium members. That the ownership structure is still being worked out, but it's fair to say that our economic interest in this consortium is quite significant. And to the extent that we're successful in the commercialization, could be a major revenue driver for us as early as 2023.
Is there a timeline on the universal flu vaccine, PDS 0202, and the preclinical data? Any expectation for timing of the release?
Well, I'll speak to the current preclinical stage that we're in. As Frank highlighted earlier in the presentation, we've received NIAID funding from the Civic Flu Program, specifically targeting unique flu antigens that are designed to be able to induce not only antibody responses, but T cell responses as well. We have confirmed that they induce these antibody responses, and testing is being done and performed by our collaborating colleagues to determine and confirm that these antibody responses actually neutralize multiple variants of the flu strains that have occurred in the last 5 to 10 years. It is still in the preclinical stages. We are aggressively pursuing funding opportunities to allow this to proceed to human clinical testing once the formulation is optimized and complete. Frank, you want to add anything else?
No, I think you've touched on the main point. So the goal here is that testing is actually very actively ongoing, both at our collaborator at the University of Kentucky as well as the NID Civic Center. And so our hope is that preclinical testing and development will very likely be completed by the end of this calendar year. And then the next step would then be to take it into human clinical trials. But really what we are focusing on today is making sure that this is actually a universal flu vaccine and that it actually addresses multiple variants and strains of the flu. And those studies are currently ongoing.
And another audience member would like to hear a description of the PDS 0203 antigen in greater detail, and wants to know if it's a hybrid protein and if it's derived from 2 viral proteins.
No, so our current antigen that's being used is not a hybrid protein. So what really is unique about our approach is really the ability to include in the viral construct, the antigen construct, conserved regions or conserved epitopes, what we call sequences of the protein that remain unchanged as the virus continues to mutate. Now, based upon Versamune's unique ability to present these proteins into the right processing pathways to train T cells, what we are seeking to achieve with this vaccine is the ability to induce neutralizing antibodies, but very importantly, train the immune systems and the T cells to recognize those non-mutating and conserved regions within the antigen, so that the patient can continue to generate a strong T cell response, including a memory T cell response against those conserved regions of the protein. So one of the things that has come up, the pandemic has continued to mature and grow. One of the emerging data actually is showing the relative importance of CD8 T cells, or T cells, and CD8 T cells in particular. And also very recently, the fact that these T cells are able to recognize multiple variants of the SARS-CoV virus. And that's really what we are designing our vaccine to do. It's not only to induce neutralizing antibodies to the spike protein, but very importantly, to recognize those conserved epitopes and regions in the virus that will allow the patient's immune system to recognize and protect against a broad range of variants as new variants continue to emerge.
Another audience participant would like to know if there's any active preclinical work with the TARP antigen, and if so, when would data be presented, and if we can look forward to a phase 1 trial at any time in the distant future.
I'll take that question because I have a particular passion for TARP, and thank you for asking that. We have advanced our preclinical work with the TARP antigen. This is actually a novel tumor antigen that was discovered by the NCI. We think it has potentially tremendous potential as an immunotherapeutic because TARP is overexpressed in approximately 90% of breast cancers, 90% of prostate cancers, 50% of breast cancers, and actually was just reported last year to be expressed in 100% of tumors from both pediatric and adult patients with acute myelogenous leukemia in the marrow of individuals resisting that cancer. So it's an important tumor antigen. It's already actually been tested in human beings, not the formulation that we currently have, but formulations using 2 small pieces of the TARP protein have already been tested in humans, in men with prostate cancer who had what we call PSA biochemical recurrence. So their prostates were taken out.
Lauren, you may want to add that you led that clinical trial.
Well, I'm not about tooting my own horn, but yeah, I was the one that delivered it and ran the trial and so forth. But the more important message is that these 2 small pieces of the TARP protein were highly immunogenic, well tolerated. They induced immune responses, exactly recapitulating what we saw in preclinical animal model studies. But importantly, it actually slowed the rate of tumor growth as measured by the PSA and actually reduced the rate of PSA rise by over 50%. This again was with only 2 small pieces of the TARP peptide, and it was in a very restricted population of men with prostate cancer. Our current PDS-0102 formulation actually has 2 very long peptides, 38 and 32 amino acids in length, that cover the entire TARP protein, which allows it to be delivered to men of every HLA haplotype and race, and allows us to induce an immune response that's very broad. Specifically, we have seen in our preclinical animal model studies induction of TARP-specific responses that were 4 to 5-fold higher than that which was observed in human clinical trials. So we are really in the very late stages of formulation of the TARP vaccine and are actually seeking non-dilutive funding as well as partners to proceed with testing this 2nd-generation TARP vaccine in humans. The goal would be to do that, to obtain that funding by the end of this year and proceed into first-in-human testing sometime in 2022. And we are very excited about the potential not only in prostate cancer, but also breast cancer. A reminder to investors out there that the most successfully exploited tumor antigen in breast cancer is HER2, but that's expressed only in approximately 25% of cancers. So we believe the potential for TARP in breast cancer is equally at least as promising.
Okay, well, thank you all for a very interesting discussion and all of the new insight and understanding about what you're doing at PDS. This is all the time we have for today. So if any of the audience members would like to get some of the independent research, they can go to Channel Check and download any of the company reports that we have on the website. Again, thanks to our guests from PDS, Dr. Bedu Addo, Dr. Wood, and Dr. Van Voorhees. I'm Robert LeBoyer and can be reached at Noble if there are further questions. Thank you very much.
Thanks a lot.
Thank you for having us.
Thank you for joining us for this virtual roadshow presentation brought to you by Channel Check. View our YouTube channel for more video content, including C-suite interviews, virtual roadshows, and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and 6,000 other small and microcap companies.